Today's lesson is all about genetic crosses. So before we go into what the genetic cross is and what it's all about and how you guys should be able to do it, I first just want to focus your guys' attention on this man right here. So his name is or was Gregor Mendel. He lived in the 1800s. He was an Austrian monk and he is known as the father of genetics. So I'll explain now quickly what that means, but basically what he did is in his free time in the monastery, he actually dabbled breeding with plants and specifically with pea plants, hence the picture. So usually if you see a pea plant, it's associated with him. So just quickly what he did is he realized that if he crossed a specific two different traits, so in the pea plant he looked at a couple of things, but the first thing he looked at, let's say for this example, was height. So he realized that if he takes a tall pea plant and he breeds it with a small pea plant, height-wise, it can give you, usually the offspring in this case, a tall plant. Okay, so just ignore this side for the moment. If you've got a tall individual mated with a small individual, these plants in the first generation is called the P1 generation or the parent generation. Whether this one is the male and this one the female or vice versa, doesn't matter. But if you have a tall plant and you mate it with the same species, a small plant, he actually gets tall plants. So not just one baby, but meaning all the babies then would be tall. Then what he did, he took two of the same offspring, so meaning if this one was male, this one is female, but they're brother and sister, he mated them with each other. So it's now the offspring, and also known as the F1 generation, the first filial generation, so it's the offspring. And if you mated them with one another, then he got in the second generation, so meaning this is now the grandchildren of these two, he realized that some came out tall. Usually this is percentage wise, meaning not 4 children, but let's say, okay, for argument's sake, let's say there were 4 offspring that came from this crossing, then 3 of them would be tall and 1 would be small. So point being, he dabbled in this characteristic, height, and then he realized based on who he crossed with who, he got specific results at the end. So he dabbled in this basically mainly with plants. I think he did do it with other organisms as well, but he mainly had his pea plants with him. And he saw, based on what he bred with what, he got specific results here at the end. So for that reason, he's known as the father of genetics, because he was the first one. You didn't know that DNA is the thing that actually dictates how characteristics is transferred from one generation to the next. He didn't know this, but he did realize that characteristics can be bred with. So if you want specifically tall plants, you had to breed tall and tall ones with one another to get tall offspring, that type of thing. Or if you just wanted small pea plants, you had to cross 2 small ones with one another to get small pea plants. So he was the first one to figure out that characteristics are transferred from one generation to the next. Okay, so Gregor Mendel, hy was 'n monnik in die Oostenryk, en waarmee hy basies daar besig gehou het, of een van sy hobbies was, om met hierdie ertjieplantjies te teel. So hy het gesien dat, gebaseer op watter tipe karaktertrek jy wil hê. Hier die een van die karaktertrekke waarna hy na gekyk het, was hoogte. So hy het sê maar 'n hoë plant gevat, 'n redelike lang een, het hy laat teel met 'n kort enetjie. So weer eens dieselfde spesies gebruik, maar hy het net verskillende hoogtes daarvan gevat en hy het net een karaktertrek op 'n tyd gekyk. So in die case het hy net gefokus op hoogte. En toe sien hy maar 'n hoë een of 'n lang een wat geteel is met 'n kort enetjie, gee vir jou lang plante. So in hierdie geval, hierdie is wat hy nou gekry het uit daai kruising uit. En so het hy 'n klomp van hierdie kinders gekry, wat so lank is. En toe kry hy 2 van dieselfde kinders. Meaning dat hierdie is broer en suster, mannetjie, wyfie. Maar van dieselfde kinders van die vorige kruising, die nageslag, het hy toe gekruis met mekaar. En toe die kleinkinders, wil ek half sê, van die eerste kruising, toe sien hy dat gewoonlik 3 uit 4 keer kom hulle groot uit of lank uit, maar dan 1 uit 4 keer kom hulle gewoonlik klein uit. So hy het nie regtig geweet hoekom dit gebeur nie, hy het geweet dis DNA wat veroorsaak nie, maar hy het besef dat dit is iets wat jy kan beheer. Gebaseer op hoe jou ouers lyk, sal dit vir jou bepaal wat jou kinders na sal lyk. So hier het hy net gekyk na hoogte en toe sien hy, okay, bepalende of hy nou wat hy met wat kruis, gee vir jou 'n sekere hoeveelheid hoë of klein plantjies in die nageslag. So F2 generasie hierso verwys na die kleinkinders van die oorspronklikes. So P1 generasie is jou parental, so dis eintlik jou ouer generasie. Dan kry jy jou nageslag, wat gewoonlik die F1 generasie is. Ons noem dit P2, as jy hulle nou weer gaan vat as die ouers. So hierdie twee wat nou gekry is, al is hulle broer en suster, word gesien as die ouers van hierdie hieronder. Okay, so guys, just quickly what I did not mention is that F1 generation, your first filial generation, this is your offspring, yes. But if you do cross brother and sister with one another, again, these are seen as the new parents for the next crossing. Then it's known as P2 generation, meaning the second parental generation. Then that gives you a F2 generation, so the second filial generation. So it's the grandchildren of the first crossing. Okay, so then I quickly just want to go through some terms with you guys. So the first one is the monohybrid cross. That's basically what the previous slide is showing us. So a monohybrid cross is when one characteristic is investigated. So mono means one, hybrid means a crossing. It's a crossing between the same species, but we're looking at different characteristics. Different variations of the same characteristic. There's a difference. So one characteristic we looked at, in this case height, is investigated. So it's a monohybrid cross. One thing we're looking at. So this is basically what Mendel did. He just looked at one characteristic at a time, and he's known for his monohybrid crosses. So your monohybride kruising is basies net wanneer een eienskap ondersoek word. In hierdie geval het Mendel net gekyk in die vorige slide na hoogte in die plante, net hoogte. Ons het nie gekyk na hoogte en kleur en blommetjie, shape en so goeders nie, ons het net gekyk na hoogte, so dis 'n monohybride kruising, mono beteken een, so dis een karaktertrek waarna ons kyk. And the second thing here says inheritance pattern, so in this case the inheritance pattern, the way it was inherited from one generation to the next, is called complete dominance. So complete dominance means that usually the one variation of a characteristic is more dominant than the other one. So in the previous slide we saw that most of those plants that came out were actually tall. So the plants came out tall, no matter the parents. So that means tall is dominant over a short or a small plant. So when you're looking at your characteristic, height, tall is dominant in the previous slide over shortness, or shortness. So basically it's when one allele masks the effect of another. So your dominant allele masks the effect of the recessive allele. So the recessive one is the one we generally don't really see, the phenotype we don't really see often in nature. So the dominant one is usually, if most plants are usually tall, that is your dominant one. So tallness then would be dominant, but shortness of a plant would then be recessive, so meaning it's rare. We rarely see it in the wild. Okay, so jou oorerwingspatroon hier van 'n Mendel baie gekyk het, was jou volledige dominansie. En dit beteken dat gewoonlik een alleel verdoesel die effek van 'n ander. So in hierdie geval wanneer ons gekyk het na die hoogte van die plante, gewoonlik die langer plant is dominant oor die korter plant. So dan sê ons die hoogte, die feit dat hulle vir die hoogte die lang plant, so die lang plante is dan dominant oor die recessieve kort plantjie. So die wat ons altyd nie wilt sien nie, word dan recessief genoem. Hy's baie rar, ons sien hom nie altyd nie. Hy kan tevoorskyn kom, maar gewoonlik in die natuur dan is hy rar. So dit is jou recessief, ek dink R rar, R recessief, sien ons nie altyd nie, maar die wilde tipe, die een wat ons gereeld sien in die omgewing, in die geval vir hoogte was dit lang plante, dan die lang een gedeelte is dan die dominante een. Okay, then thirdly is Mendel's law of segregation. So again, he did not realize again this was about DNA at the time, but this is now coined his law, because we talked about previously with meiosis in the previous lesson that usually characteristics are controlled by two alleles. So meaning again you've got your chromosome, one from mum, one from dad. Each one has a gene on it that has to do with, in this case, let's say height. So you've got a height gene in your individual, on the chromosome that comes from mum, and also a height gene on the chromosome that comes from dad. Each individual has it. You have it, I have it. any plant has it, animals and so on. So there's always 2 chromosomes together with 2 different alleles, but the same gene. Okay, so usually the characteristics are controlled by 2 alleles that separate into different gametes during meiosis. This is why it's called the law of segregation. So again, during meiosis, your alleles will separate from one another, each going into a different gamete. So this is what it means, the law of segregation, but it's referred to as Mendel's. So what happens then is when the gametes form, and when they, I want to say, come together again later on during fertilization, that will tell you in the next generation whether this individual will be homozygous or heterozygous. Okay, so in the next slide, this will become a bit more apparent. Okay, so jou Mendelse wet van segregasie is basies die eienskappe word beheer, enige eienskap, beheer deur 2 allele, wat dan skei van mekaar na afsonderlike gemete tydens meiose. So onthou terug in meiose, wanneer ons gaan na metafase, dan is die die chromosome gewoonlik gerangskik op die ewenaar, maar hulle gaan skei van mekaar. So dis weer daai krake skeiding wat gebeur. Maar die punt is die 2 chromosome, een van ma, een van pa, en elkeen het 'n geen op wat op 'n spesifieke alleel is. So daar's 2 allele vir 'n spesifieke geen vir 'n spesifieke eienskap, in hierdie geval nou hoogte. En dan word hulle van mekaar geskei wanneer die gamete vorm. En onthou word nou van diploïd na haploïd. So omdat dit dan skei, dis waarna hierdie wet verwys. So elke gameet gaan dan net 1 geen hê of net 1 alleel hê van hierdie geen in hom. Okay, so dit sal 'n bietjie meer obvious raak in die volgende slide. Okay, so then just quickly, this one shows you guys just quickly some different characteristics that Mendel actually looked at. So he looked at a couple of, he realized some things are dominant. So some of the traits are dominant, some of them are recessive, like I just said. In the wild, we usually see this variation, or he saw this. And then usually in the wild, some of these we don't really see it. So they're recessive. The rare ones are known as recessive. The ones we normally see in nature is the dominant ones. So Mendel, he dabbled with flower color. He looked at the plant height. We just had in the previous example. He looked at the seed color of the peas, the seed shape, pod color, pod shape, flower position, all those things. So every time he bred his plants, he looked at one of these things at a time. So then he realized something like purple is dominant over white. So normally the plants or the flower color of a pea plant will be purple and not white. Usually the plants will be tall, like we just saw in the previous example, and normally they're not really short. It's very rare for them or recessive of them to be short, and so on for the other examples. You guys don't have to memorize this slide. This just demonstrate that he realized some things we normally see in nature, some things we rarely see in nature. So die dominante eienskappe, teenoor jou recessieve eienskappe, die recessieve ene reg hieronder, verskillende kleure, soort kleure, van die blomme, die hoogte van die plant, die kleur van die saad en so aan verskil, van tyd tot tyd, en het agtergekom dat hierdie eienskap hieronder is een eienskap, maar verskillende variasies. Hierdie ouens hieronder is recessief, baie raar om te sien, en party van is dominant, soos wat ons hierbo sien. Okay, so then I just quickly want to talk about the genetic cross, and just very quickly. So I think I will go on with this in the next lesson. But basically what this one is all about is, firstly, like that first slide we had, we saw there was a P generation. This is technically P1 generation, so your first parental generation. So in this case, normally one is male, one is female. In this case, we don't know which is which, doesn't matter. Let's make this one male and this one female. And this X represents the fact that they're going to cross with one another. So in this case, we're just looking at one characteristic. And in this case, it will be again the height of a plant. So just looking at height. So usually, height is represented with 2 T's, but 2 capital T's. Why 2 T's? This represents one allele, let's say from dad, and this represents one allele from mum. Okay, so usually because it's just one characteristic, we write the letter twice. So it's T. I'm assuming T stands for tall. So in this case, a capital T means that this guy is a tall plant. And again, why 2? Because again, it's 2 alleles. So meaning this individual is homozygous, but homozygous dominant. Okay, so it's 2 capitals, A capital T represents a tall plant. We can see in this picture it's tall. And it's homozygous. Why? Because there's two of the same alleles, one from mum, one from dad. Okay, so if we were to ask you what is the genotype of this plant, you'd say it's homozygous, dominant. Why dominant? Because usually the capital T represents the dominant one. Okay, so always when it's a capital, it's dominant. In this case here, we've got a shorter plant. We can physically see it's a shorter plant than this one. And the genotype, right here, what the alleles would be, it shows us it is also homozygous. Why? Because there's two T's that are the same shape and size, and two of the same type of T's, but they are small T's. They're not capital letters. In this case, it's capital, but in this case, it's small. So again, why do we still work with T? The T shows us that we're working with the same characteristic. So capital T means is a tall individual, so it's height, a tall individual. But here we still have to talk about a T because we're still working with height, the height of the plant. But we say it's a small t, we write it as a small t because this guy is a small plant. Okay, so we don't use an S now to represent small. We don't say T and S because we want to indicate it's the same characteristic. So it must be a T, same characteristic, but remember different variations. This one is tall for height. This one is small for height, or short for height. So two small t's. So this one then is homozygous recessive because we normally don't see this one in nature. Okay, so they in a question and so on, they will tell you which one is dominant usually and which one is recessive. So again, what recessive just means is in nature we don't always see the short plant. Usually your pea plants in nature are tall by nature. So this one's the dominant one, this one is recessive. Okay, so then usually what happens is the gametes form. So why do they show this as one T? Because again, remember during meiosis, this guy is going to separate from one another. This was on one chromosome. This one is on a second chromosome. So these two physically split from one another. So in this case, this picture doesn't show the two gametes that form because usually it will form two gametes. But because it's going to be two capital T gametes, they just didn't write both. They just wrote the one. Okay, don't get too confused with this. In one of the next slides or in the next lesson, I will show you guys usually how we write it in an exam example. So I just want you guys to realize that this represents the gamete. Okay, so you usually make a circle around it just for yourself. So you remember, okay, this represents the little gamete. Whether it's the egg cell or the sperm cell, does not matter in this case. Just make a circle around it so you remember, okay, this is the formation of the gamete. And only one of these alleles go in there. Why? Because of the law of segregation. After this, these will split from one another because meiosis will happen here. So from this area until we get the gamete, meiosis happens because why? Meiosis causes gametes to form. Okay, same thing happens here. Meiosis happens in this step. And then these two alleles separate from one another. So we're going to have two of these gametes technically with one allele in each. Okay, so again, we're not showing all the different chromosomes in the gamete because we are just focusing at this point just on this characteristic, just for height. So we're just looking at the allele for height. Okay, so then in your F1 generation, what happens? The egg cell meets the sperm cell, and then we have fusion, fertilization. And now in the next generation, the F1 generation, the offspring generation, so to speak, we have a tall plant. And what are the alleles? Capital T meets small t. And now we have this for the genotype. So we can see it's a tall plant. Why? Because it has one capital T, and this capital T is dominant over the small t. So physically, the phenotype, we can see it's a tall plant. But genetically, the genotype will then have a capital T and a small t. So this individual is not homozygous. This individual is heterozygous. Hetero meaning two different alleles for the same characteristic. Okay, hope you guys are with me so far. So if we were to, hence the Punnett here, or this block, what it represents, if we were to allow this guy to mate with itself or to mate with its sisters or brothers or whatever, they say this is a male. and we allow it to mate with its sister that also looks the same as it. What then we do is, this is a Punnett square. In the next example, I will show Punnett square again. But if we were to mate this guy with itself, or its sister, or its brother, with the same genotype, here it represents female, this one is male. So let's say this was the male. Here's the one gamete for it. It has a capital T in the one gamete, a small t in the other gamete. So these two blocks represent the sperm in this case. It is for the male. So capital T, small t. But remember, if it mates with itself or it mates with its sister, it'll have the same genotype. So capital T, small t. And then you just add these two together. So in this block will have a capital T, capital T. In this block we'll have a small t coming down, capital T going over. So this one will be heterozygous. This one will be two capital T's, homozygous. This one then will also be heterozygous. Why? Because you put this capital T to the bottom, this small t there, and the last one will be homozygous recessive. Why? Because you've got a capital T here going down and a small t, and a small t going that way. So then we have a homozygous recessive individual. So what I want you guys to see is that if we mate two heterozygous individuals with one another, heterozygous with heterozygous, we get one homozygous dominant individual, or the chances for it actually. Okay, I'm going to say it now. Homozygous dominant one. We can get two heterozygous ones, offsprings, and we can get a recessive, homozygous recessive one. Okay, so just quickly, this does not mean that we have 4 individuals. This does not tell us the amount of offspring this organism can get. We can't get 4 offsprings from this individual or this crossing. It shows us the chances of getting a homozygous dominant one here is 1 out of 4. It tells us the chances of getting a heterozygous one is 2 out of 4. Sorry. The chances of getting a homozygous recessive one is 1 out of 4. So what this block just tells us are the chances of getting a specific genotype as an offspring. Okay. Ek gaan vinnig Afrikaans, skies. As jy vir jou P-generasie, jou ouer generasie, as jy kyk na 'n hoë plant, in hierdie geval het ons 'n homozygotiese dominante een, hier het ons 'n homozygotiese recessieve een, so hy is die raar een wat gewoonlik nie sien nie. As hulle met mekaar gekruis word, eers moet jy hulle gamete kry, so na meiose word hierdie vir mekaar gesplit. Hoekom? Omdat weer eens met my metafase gaan die een kromosoom na die een kant toe gaan van 'n gameet, hierdie na die ander gameet toe. Okay, so hier wys dit nou nie al twee gamete wat gaan vorm van hierdie meiose proses nie, maar basies die gaan twee wees, hulle gaan dieselfde lyk. Een T gaan in die een gameet in, hierdie T gaan in die ander gameet in. Omdat hulle dieselfde lyk, het hulle nou net hierdie een keer geskryf. Okay, so hier gaan dis wat die gamete gaan lyk vir hierdie individu. Hierdie is wat die gamete gaan lyk vir hierdie individu, so klein t's. So weer eens, in hierdie geval het ons 'n hoofletter T, want die ou is dominant. Ons sien dit gewoonlik in die wild tipe, in die natuur. So ons gebruik T in al twee gevalle, want T verwys na dieselfde karaktereienskap. Ons kyk na hoogte. Maar T, die hoofletter T verwys na 'n baie lang plant, en 'n klein t verwys na 'n kort plant. Okay, so dit wys na dieselfde karaktertrek, maar ons gebruik 'n hoofletter en 'n klein lettertjie om te onderskei tussen dominant en resessief. Dan het ons twee gamete. Al die gamete hier gaan resessief wees, al die gamete hier gaan dominant wees. As hierdie twee mekaar kom met bevrugting, wat hier gebeur, sien ons dat die volgende geslag, almal van hulle toevallig, lyk heterozygoties. So dit is almal lank. Hoekom? Want ons 'n dominante alleel wat die effek van die resessiewe een verdoesel. So genotypies is die ou heterozygoties, maar fenotypies is hy lank. Hy bly lank, want hy het net een van die allele nodig om lank te voorskyn te kom. So die een verdoesel die ander enetjie. Maar hy is nou genotypies, is hy heterozygoties. Nou die blok hier langs aan wys, teoreties, as ons die ou met homself sou bestuif, van plante kan dit doen, of hy het broer en susters gehad en sê maar hierdie is die mannetjie en ons laat hom teel met sy suster wat presies dieselfde lyk as hierdie van hierdie kruising. Dan wys hierdie net, hier is jou eerste gameet, hier is jou tweede gameet. En onthou as hierdie ouetjie nou split, dan gaan een gameet 'n hoofletter T hê en die ander gameet gaan 'n klein T hê. Gaan nie dieselfde wees as hierdie geval nie, want hy was homozigoties, die ou is nou heterosigoties. So een hoofletter T word dan daar geskryf, 'n klein T word hier geskryf. Dit verteenwoordig ons twee tipes sperm wat jy kan kry. Dan dieselfde gebeur vir die vroulike plantjie ook. Hier is een tipe gameet wat jy kan kry, tweede tipe wat jy kan kry, want sy is ook heterosigoties, soos jou eierselle. Dan wat hierdie vir ons wys net, is die moontlikheid. Jy het 4 moontlikhede van nageslag om te kry. Dit is nie noodwendig 4 kinders wat hulle gehad het nie. Dit wys net die moontlikheid van kinders, genotypies en fenotypies, hoe hulle kan lyk. So jou kanse om 'n homozigotiese dominante kind te kry, is 1 uit 4. Jou kanse om 'n heterosigotiese kind te kry, is dan 2 uit 4. En kyk, hier is 'n moontlikheid, wanneer hierdie eierselletjie en hierdie spermsel mekaar verbind, 'n hoofletter en 'n klein lettertjie hierso, heterosigoties, maar hier is ook daai kans. Die een van die ma en hierdie een van die pa kan bevrug word, en dan kry jy ook 'n heterosigotiese een, soos 2 uit 4 kanse vir die heterosigoties, maar ook net 'n 1 uit 4 kans om 'n homozigoties recessiewe kind te kry. Okay. Maar jy het 'n 1, 2, 3 uit 4 kans om 'n hoë plant te kry, en 'n 1 uit 4 kans om 'n klein plantjie te kry. Sorry guys, I just forgot to mention in English that with these possibilities for the genotypes, you also have 1, 2, 3 possibilities, 3 out of 4, to get a tall plant, but only a 1 out of 4 chance of getting a small plant, because no one of the other gametes, when they fuse, will give you small plants. Only this one. Only when a small t and a small t fuse together, get a homozygous recessive one. Okay, so in the next lesson, I'll go through other crosses that'll hopefully make this a little bit more clear for you.